Hello, good morning, good afternoon, or good evening, depending on where you are in the world tonight. I would like to welcome you to Less to Do with the Neuron, Morphology, and Anatomy. And we'll start right off the bat with we'll go right back to the beginning and we'll talk about the differences between the two major classifications of cell types, prokaryotes and eukaryotes. Now, prokaryotes are organisms that lack a cellular nucleus or any membrane encased organelles. Many prokaryotes also have a cell wall. Some eukaryotes also have a cell wall, but not as many. That's the exception rather than the rule. Now, the interesting thing is about the eukaryotes. So humans are multicellular eukaryote organisms, and the membranes that we're talking about, or the organelles that we're talking about that have membrane structures, are things like mitochondria. So the mitochondria are making ATP inside the eukaryotic cells. Now, both types of cells have ribosomes, which make proteins. And the major difference is that the enclosed nucleus contains DNA material in the eukaryotic cell. And inside the nucleus, there is also a nucleolus, which has highly defined nucleic or nuclear compounds inside. We're going to also talk about the cell membrane structure. So the basic cell membrane structure in a eukaryotic cell or in a human cell or in a neuron, which is what we're going to be focusing on, is this bilaminar structure here. And so there are two layers of phospholipids, one layer here, one layer here. And the phospholipid layers are composed of a area that is hydrophilic or water-liking, and an area which is hydrophobic, which means that it is not water-liking or water-hating, or it's more fat, areas of fat or non-soluble with water. So the area of the hydrophilic end of the phospholipid is composed of phosphate and glycerol, with the chemical structures shown here in the slide. So we have this phosphoglycerol or phosphoglyceride attached to a fatty acid chain, and the fatty acid chains can have high degrees of saturation or can be unsaturated. And in fact, the unsaturated fats or the unsaturated fatty acids are the ones that have a series of double bonds, which causes the hydrophobic areas to have a little bit of a bend in them. You see this bend that's happening here. And that bend is important because it can separate or cause spacing to happen between these phospholipid layers, and cause them to have different attributes or different functionalities. Now, I want you to look at the big ball structure here, and I included this diagram because I think it's very interesting, because it shows kind of in a three-dimensional sense how this membrane surrounds the stuff on the inside. So we have the intracellular area, which means inside the membrane, and then we have the extracellular area, which means outside the membrane. And these are all all of these are relative to whether things are inside the cell or outside the cell. And so we're going to be referring to these a lot throughout our study, not only just of the neurons, but also of anatomy. So this is a very important construct. So we have the cell membrane, which means it's composed. Cell membrane is separating the cell from extracellular events and intracellular events. And this lipid or phospholipid bilayer is very good at keeping a separation between the things that are inside the cell and the things that are outside the cell. So it forms a barrier to control what gets in the cell and what doesn't get in the cell. And on the next slide, we'll talk about how the barrier is modified or specialized in neurons and in other cells to move things across this barrier efficiently or keep things out efficiently. So to summarize, we have the phospholipid. So this phospholipid composed of phosphoglycerol at the top, which is hydrophilic, and the fatty acid chains at the on the bottom, which are hydrophobic in nature. And they form a bilaminar structure, which means two of these phospholipid layers. And that acts as a barrier, keeping the inside and the outside of the cell separate so that we can have different functionalities inside and outside. Now this is now looking at the membrane structure with more specialization. And the specialization is involving the insertion of proteins into this membrane. So we have different types of proteins that do different types of things, and we have proteins that go all the way through the bilaminar layer, and we call those integral proteins. We have proteins that maybe are only on the inside of the layer, which we call peripheral proteins. And we have proteins that are on the outside that may or may not go all the way through the membrane, but they have specialized attachments or specialized chemical structures attached to them, usually formed of some kind of carbohydrate or glycolipid or glycoprotein. So these glycoproteins, carbohydrates, and glycolipids act as very specific attractors or very specific signaling components of the cell membrane for signaling different for identification of the cell. So for instance, if a immune system cell comes in contact with one of your body cells, it needs to know, is it a self or is it something else? And these glycolipids and glycoprotein structures are important in delineating that. That's just one of their functions. They have many others. So you'll see there are proteins that are going through the complete bilayer. There are proteins that are only on one side of the bilayer, and there are also proteins which we call channel proteins. And these channel proteins are extremely important because they allow things to move in and out in a specific fashion. So from the outside of the cell, inside the cell, or from inside outside. So, and these channel proteins we'll find as we learn more in later lessons, can be passive or they can be active. There can be a variety of different functions that are associated with these. We also see another very important component, component that is composed of protein, and that's these alpha helix proteins, which often span the membrane also. And they have a variety of specific functions also, which we'll get into later. Now, I just want to point out one other aspect is this cholesterol. Now, cholesterol is an important component of the fluidity of this layer. So you remember this layer, this bilaminar structure, the cell membrane, has to be strong enough to keep things in and out, but it also has to be fluid to allow the cells to move around and have contact with each other and to be fluid in nature that allows a lot of these proteins to do their function. So that cholesterol is, even though lots of times we think cholesterol is bad because we're thinking about cholesterol in the bloodstream, okay, cholesterol in the membrane is actually an essential component. So we just remember that cholesterol isn't all bad. It has very meaningful and necessary functions when we're talking about cell membranes. Now, moving into the actual cell itself, we are going to just look at how these, or what some of these different organelles are that we talked about that were in the eukaryotic cell. So here's the nucleus that we talked about. And so this is a separate membrane, okay? Remember, this is the cell membrane out here that we just looked at. And then inside the cell, there are other membranes. So there's a membrane here, which is a very specialized membrane that's enclosing the nucleus. There are membranes surrounding the mitochondria, and we'll learn about these in future lessons also in a lot more detail, but there are membranes surrounding the mitochondria, and then there's membrane structures known as the Golgi complex, and the smooth endoplasmic reticulum, which are involved in transporting a variety of things throughout the cell, modifying chemical structures. There's also these vesicles, which will join the membrane and expel different substances into the extracellular space. So this gives you a three-dimensional understanding, or a three-dimensional picture of what's happening inside that cell. And these are some of the organelles that are now surrounded by a membrane or encased in a membrane, which is not what happens in the prokaryotic cell. Now let's talk about, again, let's look first of all at the generalized neuron here that we have. And this generalized neuron has a cell body, and inside the cell body it has these different organelles, which we're going to learn about on the left. And it also has this specialized structure called an axon. And we're going to learn a lot more about this axon as we progress through this module. So I just wanted to give you an idea of how these organelles sit inside the neuron. And now if we come over here, we'll see that what are the different organelles. So this is the cell surface bilaminar membrane that we've already talked about. And obviously, as we've discussed previously, it controls passage of some entry of substances, exiting of substances from the extracellular or intracellular space. It also is very important because it is the site for receptors. So remember those glycoproteins and those glycolipids that we talked about that were on the surface of the cell? A lot of those are intimately involved in receptive functions. So receptors for hormones, receptors for neurotransmitters, a lot of very important things that receptor function will help. Now, the nucleus is the place where the DNA is stored, and it has little passages or it has proteins that are basically what we call nuclear pores, which allow mRNA, okay, so messenger RNA, and ribosomes to pass through the membrane and go in and out of the nucleus, which is very important in expressing genetic activity. And we also have mitochondria, which I briefly touched on before. And the mitochondria carries many functions. Probably one of the most well-known functions of the mitochondria is the production of ATP. Now, the mitochondria is also important in a lot of other aspects of cell function. We'll get into more details with that as we progress through the program. I just want to give you a generalized overview of these organelles right now so that you have an idea of what we're talking about when we start referring to these things. Now, lysosomes are basically digestive. They're vesicles that, or containers that hold digestive enzymes that are used to break down pathogens or that get inside the cell, or perhaps old, worn out cell structures, like old organelles that need to be dissolved or need to be removed from the cell. So the lysosomes are important for maintaining those types of sort of maintenance activities that have to happen inside the neuron. Then we have the ribosomes themselves. Now, the ribosomes are where the RNA, so the RNA that contains the genetic instructions from the DNA bind, and this is where the actual proteins that we were talking about are produced. So the RNA contains the genetic code from the DNA. It binds to the ribosomes, and then the ribosomes are programmed to construct proteins that are necessary for basically all of our survival, because proteins are used in a lot of different areas inside any cell, but especially in the neuron. Now, there are two different types of endoplasmic reticulum. Now, one type of endoplasmic reticulum, which is called rough endoplasmic reticulum, is an endoplasmic reticulum that actually has ribosomes intimately attached to it, or involved with producing proteins that need to be inside another membrane as they're produced. Now, the smooth endoplasmic reticulum is, excuse me, is important in the transfer of lipids and carbohydrates. Okay. Now, the Golgi apparatus basically takes proteins that have been produced and it modifies them, and it can package them for export or package them for transport throughout the cell. Occasionally, you will see movement of proteins from one part of the cell to the other. In neurons, a lot of movement happens up and down the axon, and we call that axoplasmic flow, and it goes in both both directions, up and down the neuron. In generation of action potentials, which we'll talk about also the next few lessons. But this axon comes down and it flows all the way down, and these axons can range in length from a few millimeters to a meter and a half in some people. So, for instance, cells coming from your spinal cord down to your toes, maybe in some people as much as a meter and a half in length. So these axons can vary to their length, and they also vary in whether or not they have branches. So in this case, we have an axon coming down, and off that axon comes another axon, which we call a collateral axon. And this collateralization of axons means that the contacts that a single neuron can have can be as many as 10,000 other neurons. So now if we go back, and in fact sometimes in the cerebellum can be as much as 50,000 other neurons that they contact. So if we go back to the 85 billion, now we times that by about 10,000, we see there is a lot of contacts inside the brain. So that's the complexity that we're dealing with. Now this axon goes all the way down, and it ends in what's called a synaptic structure. Okay, so a synaptic terminal, axon terminal, whichever you prefer to use. And at the end of these axon terminals, the process or the projection of one neuron will come very, very close contact to the projection of another neuron. So the axon of one neuron comes very close to the dendrites of another neuron, and they're separated by a little space, and that space is called a synapse. Okay, so that's pretty much the outline of a typical neuron for you. Now, the neurons can come in different morphologies or different models, I suppose. And if we look at, for instance, the different type of sensory neurons, so the neuron that we just described to you was this one over here, and this is the typical sort of neuron that people will talk about. This is usually the one that people are thinking about when they're talking about neurons. And here's the dendrites again coming off the neuron cell body. Here's the axon covered with the myelin sheath. And down here is the axon terminal, which will form the synapse with some other neuron or some other structure. If we go over here now to the sensory neurons, you'll see these are a little different, because here the body is separated from the axon terminals by the axon, where here we see the axon terminals, okay, are actually flowing in the same structure. So we call this pseudo-unipolar. Okay, so that means that the cell body is basically adjacent to the axon, and it can cause, and it has basically by definition or by convention, the part of the pseudo-unipolar axon that is bringing the receptor, so up here at the dendrites, bringing the receptor part in, is called the receptor axon, and this would be the effector axon. So this would be, when we talk about efferent and afferent, this would be the afferent axon, this would be the efferent axon. So axons, when something is bringing information towards the cell body, we call that afferent. When it's taking it away from the cell body, we call it efferent. Now, if we look now at a different type of cell, this is a bipolar cell. So this cell basically has the same type of structure as a pseudo-unipolar cell, except now the receptor dendrites and the axon are actually separated by a cell body again, but the extensions or the distance that these are away from the cell body are a lot greater than they are in basically a multipolar efferent neuron. Now let's look at some other types of neurons. Now this neuron here is basically anoxic. It doesn't have an axon. Okay, so it's all dendrites in this neuron, and all the dendrites are focusing into the cell body. We have a different other type of cell. This is what I was talking about by cells that have up to 50,000 connections with other neurons, and we see that these dendritic trees. So this is a dendritic tree. This is a dendritic tree. These dendritic trees can be massive in some neurons, and cause massive integration or collect a massive amount of information from other neurons. Now let's look at some of the differences between the central nervous system and the peripheral nervous system. And we're just going to talk right now about the differences in the myelination of the axons that occur in the peripheral nervous system and the central nervous system. So the central nervous system basically are neurons contained within the brain and the spinal cord. As soon as the neuron goes outside of the spinal cord, we call it a peripheral neuron. And the peripheral neurons have myelin. Some of them have myelin, and the ones that are myelinated with a specialized type of cell called the Schwann cell. And in the central nervous system, you'll see that the myelin is produced by a different type of cell called an oligodendrogliocyte. And one of the main differences is that in the oligodendrogliocyte, you'll see one cell is actually making contact with more than one axon. So it's forming the myelin on more than one axon, whereas if we look at the peripheral nervous system, the Schwann cells are only in contact with one neuron. Okay, now we also have, this is a very good slide because it's showing us the relationship between these big neurons and the astrocytes, or one type of glial cell. So the astrocytes are connecting the blood supply that's flowing through the brain to the neurons. And we also are seeing these microglial cells. So these little microglial cells are basically the immune system, or part of the immune system, inside the central nervous system. So again, we're going to find that the immune system has differences between the immune system outside the brain and the immune system inside the brain. Now, there's one other type of cell that I would like to introduce you to now, and that is these ependymal cells. Now, the ependymal cells line the ventricles of the brain, and these ependymal cells are basically the cells that produce all of the different cell types that are in the brain. And when we talk about the embryology and the development of the brain in the next module, we'll get into this in a lot more detail. Now let's talk now about what we talked about in the peripheral nervous system. The Schwann cells are the cells that are forming the myelin sheaths. Schwann cells and oligodendrogliocytes both form myelin sheaths, one in the peripheral nervous system, one in the central nervous system. Now, interestingly enough, the Schwann cells can also release neurotrophic factors. Now, neurotrophic factors are factors that cause neuron growth or inhibit neuron growth. So sometimes the neurotrophic factor can do both, or either or. And there's also some satellite cells in the peripheral nervous system that provide support for cell bodies in the peripheral nervous system. But if we go into the central nervous system now, we see that we have a variety of different glial cells. We have the astrocytes, we have the microglia, we have the ependymal cells, like we've already talked about. And so astrocytes are very important. They form a lot of different functions inside the brain. They provide some general support, so structural support for neurons and other cells and structures inside the brain. They also help form the blood-brain barrier, which is a barrier that keeps toxins and unwanted components out of the brain. And we'll talk about that in a lot more detail later also. This is just a general introduction, just so that you can get an idea of how things are, or what cells are involved in what. Now, they also secrete neurotrophic factors. So in the central nervous system, astrocytes are important in that. In the peripheral nervous system, the Schwann cells are important in secreting those things. And another topic that we're going to talk about in this module is about neurotransmission and neurotransmitters. And one of the other functions of astrocytes is that they will help reuptake or remove neurotransmitters from synaptic areas so that cells don't get overstimulated by these compounds, which is sort of all put together in the homeostasis of how the nervous system works. Again, this is a diagrammatic representation of how the ogliodendrogliocytes and the Schwann cells differ. And what I'll introduce you also to now is some different structural stuff on the axon, and those are the nodes of Ranvier. So these nodes are areas where on a myelinated nerve there is an area of unmyelination, so there's a little space between the areas of myelin, and these are important for the conduction of nerve action potentials down the axon. And we'll show you in great detail in the next few lessons how this unique and amazing structure of the myelination of these axons allows that to increase both the speed of conduction along the axon, and also how size of the axon and the myelination of the axon can both contribute to the speed of transmission of those action potentials up and down the nervous system. Now, the final thing that we're going to talk about today is just an introduction to the synapse. Now, the synapse is the specialized structure at the end of the axon, or it is where the axon will come in close contact with another neuron, usually at a dendrite of the other neuron, but as we'll see later, there's different types of synapses which we'll go through. So let's just look at the big picture over here first, and we can see that a dendrite can synapse with a number of different synapses all at the same time. So all of these yellow things are axons coming in from different neurons, and they're synapsing on this dendrite. So they can produce all kinds of different modulations in that dendrite. So what we call the neuron or the axon that is causing the stimulus or coming in contact with the cell here, producing the stimulus, we call that the presynaptic axon. And the receiving or receptor area we call the postsynaptic axon. So presynaptic, postsynaptic. So let's go over here and we'll look at one of these synapses blown up a little bit. So here is the postsynaptic area. So this is over here. So this is showing us this is the dendrite. So we mark this as a dendrite. So this is the dendrite, and on the dendrite we have receptors. And remember, these receptors are formed from that bilaminar membrane through proteins and glycolipids, glycoproteins that are on the surface of the cell. So you see how we've built from the structure of the membrane all the way up now to how this specialized structure can receive neuron transmission or neurotransmission from a presynaptic neuron here. Now inside the axon we have these vesicles full of neurotransmitter and other things, but in this case it's neurotransmitter. And they, when in the right environment, when the right stimulus happens inside the axon terminal, these vesicles will bind to the membrane and release their contents into this small space called the synaptic cleft. Now, the contents are usually neurotransmitters, which are these blue dots, and they then diffuse across this membrane and interact with the receptors that are designed to bind them. Okay, so when we have the neurotransmitter released, it moves across the synaptic cleft and it gets bound by the receptors, and then that will cause changes in the postsynaptic neurons' ability to let in different ions or keep out different ions. depending upon what effect these transmitters have on that membrane. And that will result in changes in this postsynaptic neuron, which may result in another action potential down the chain. But it's also important to realize that this neurotransmitter, once it's released, not all of it gets taken up on these receptors. Some will diffuse away, and some will be uptaken by those astrocytes that we talked about that are sitting in close contact around these synaptic structures.